Thank you so much, Ivan, for the nice introduction. Thanks also, James, for the invitation. And in order to prove, let's talk about my dark side. I'm going to have a criticism. I'm going to make a criticism to James, to the organization, which is perfect otherwise. But there's a minor criticism, which is that in this beautiful network here, that is used for the meeting and we have in the front of our program, there is one word that maybe, in my opinion, is missing, which is noise. He talks about dynamics, oscillations, stability. So at the end of the day, at the end of the talk, hopefully I can convince you that maybe noise is worth enough, is worthy of being in this type of network. So that would be the idea. That's my criticism, so thank you. Okay, so, well, also regarding the wobbling, probably the problem is because it's periodic. If it was noisy, it would also be better for us. So. And that's the whole idea. So what I would like to discuss today is a little bit of the work that we have been doing in recent years together with many other labs, trying to show that noise could also, it's not necessarily something bad, it's something that maybe cells can use for their benefit and in different kinds of biological control networks. So the idea is the following. I mean, if you think about it, irrespective of whether you think of the universe in terms of or if you have a mechanistic view of the universe, or if you have a view which is based on the indeterminacies of quantum theory, there is one clear thing which happens, which is that when you have many degrees of freedom that are interacting, you're going to have stochasticity and randomness. Classical example, for instance, is Brownian motion, where the irregular motion of these little beads here is due to the random collision of many molecules of water that are acting on these beads, and then you see all this source of noise. Now, of course, cells are much more crowded than a fluid. So it is natural to expect that you're going to have many opportunities for noise to arise, and in fact, that is the case. If you just culture a collection of isogenic cells, you're going to see that in different conditions, you're going to have large variability, not only cell to cell, but also in time. And the question is, how do biological systems cope with this type of variability and this type of noise? And of course, there are two options. You either lose the noise or use it. And of course, the first thing is, oh, noise is bad, so let's try to see what cells do in order to avoid having to cope with noise and trying to filter out. And in fact, some people in recent years have been proposing mechanisms. For instance, Alfonso Martinez Arias a few years ago was proposing that beta-catenin was controlling the level of stochasticity by somehow modulating chromatin and stabilizing chromatin state. But of course, it is hard to believe that the cell, I mean, because this is one particular case, but it's hard to believe that cells have widespread noise filtering mechanisms because it would be too costly. So another interesting and appealing hypothesis is that, at least in part, cells are using this type of intrinsic stochasticity to do something useful. And this is what I would like to talk to you about today. In order to show to you that this can happen, because one could think, oh, how is this possible? If noise is something completely disordered, how can get something ordered out of it and something coherent? So I want to make a psychophysical experiment with all of you. Some of you probably have heard me about this already. And I'm going to show you, what I'm going to do is I'm going to show you an image with a very low contrast. And I want to ask you to try to identify the image. You may be successful already, but let me show you the image. This is the image. Okay, the contrast is maybe too low to see what's in the image. So what I'm going to do is I'm going to take each one of these pixels and I'm going to add just a little bit of a random number with a zero mean. So it could be positive or negative. So I superimpose on this some kind of random number. Now, if I overdo it, if I put a lot of noise, what's going to happen is that you're going to have also a very complicated image. And you can probably already see what's going on here. But the thing is that if you don't add so much noise, you just add a little bit, then you have a beautiful view of the image, especially for the British, and you recognize what's in here, okay? So this is a very simple and very simplistic example of how can you can use noise for your benefit, depending, of course, on the problem that you are posing, okay? And now you might think, oh, this is very complicated because this involves perception and interpretation of the image, but in fact, if you look at the activity of the visual cortex in this case, it is true that when you add a little bit of noise, the activity in that part of the brain is enhanced. Okay, so you can also see structurally, you can also see at the functional level the effect of noise in this type of behavior. Now, how does this happen? The explanation for this effect is very simple. You just have a signal which is carrying information, and it's below a threshold. Since it's below your detection threshold, you're not going to detect it, except if you add some noise. Now, you can add noise to the signal, and if the noise is unbiased, and you will see that you have noise everywhere, but of course, when you are closer to the threshold, you're going to see the signal better. Noise is really helping you. So you are extracting whatever spatial or temporal frequencies you have in your signal due to noise. So noise always helps at the beginning. Of course, eventually, if noise is too large, you are swamping the signal, and you see nothing else. This is already very old. For many years, physicists have studied this effect. And in the 90s, this moved to neuroscience, and some people thought, okay, we have a beautiful system which is threshold-based, which is a neuron. So this means that neuronal systems should be able to use this type of behavior in order for them to operate. And for instance, there were experiments in this particular case by the group of Steve Schiff, in which they were analyzing cortical slices of a mouse brain, and they put them under both electric periodic signal and electric noise. And what they saw is that noise really helps the system, this piece of cortical tissue, to respond to the periodic signal. You have to have a certain amount of noise which is neither too low nor too high. Okay, this is maybe an ad hoc experiment. So also what other people were doing more interesting, or if you will, or more spectacular experiments, such as, for instance, the group of Frank Moss. They did this beautiful experiment at the end of the 90s, in which this is a behavioral experiment. They were analyzing the capacity of a certain type of fish, paddlefish, to detect prey. The prey that that this fish eat is Daphnia, and Daphnia emits a small electric signal. And the animal is using this signal in order to catch the prey. And what they did was something very simple. Since the signal is electric, they also used electrodes here to apply some electrical noise. And what they saw is that if you increase the amount of noise at the beginning, the animal was able to catch more prey. So again, noise was helping the system to operate, this neuronal detection system to operate. And this was done at the behavioral level. This was extended in humans, for instance. You can do this analysis in humans. Again, as I was telling you earlier in this simple example, you can look at the visual contrast detection, you can look at auditory detection or tactile stimuli detection, and in all cases, noise was seen to help. But of course, the question is, there are two limits here. You have the behavioral results, and then you have the single-cell microscopic results. How do you mix these two things? So what we did recently was look for a specific neuronal state, which has a very well-defined physiological meaning, and we were able to make measurements at the microscopic level. And this state is basically these slow wave oscillations that are known to occur during the deep sleep. Which is, here you can see contrast of this is activity trace measured from a cortical network. In this particular case, I think it was in vivo. And they compare, it was in vivo, of course. They compare what happens in deep sleep with the awake state. And you can see that in this particular case, you have just a normal random firing that you have in the spontaneous activity in the animal. And in the deep sleep, you have these bursts of activity with a very low frequency. And what happens is that the neural network self-organizes here in order to give rise to this behavior. Now, this behavior, again, is physiologically relevant, and the deeper in sleep you are, the more regular this behavior is. So the question is, where does the regularity come from? I mean, you will really need a mechanism that produces some clock here or not. And the thing is that the current understanding of this type of dynamics is such that there are two mechanisms or two main characteristics that this behavior has. First, the initiation of these events is stochastic. It's due to recurrent activity that somehow triggers the whole network to start operating after a while, after a period in which it has been silent. And then the question is, how does this event terminate? And one of the mechanisms, which seems that this would be more likely to be truth, is that this duration is somehow stereotyped, and it's due to the activation of some adaptive current. Okay? So if that is the case, this means that we have some dynamics at the population level in which the event here is stochastically initiated, but then the duration is more or less stereotyped or deterministic. Now, these are the two main properties that, for instance, a neuron has and allows the neuron to operate in the way that I was showing you earlier. So then it is nice to think that maybe what's happening here is that the background synaptic noise that the cortical network is receiving is optimizing the regularity of the up-down cycle. This is something that, at least from what we know of this phenomenon before, should happen. So the question is whether this is the case. What happens if we add noise to this system? Is this really becoming more regular, which would happen when you are going into deep sleep, or not? So the first thing that we did is we analyzed a simple, well, a relatively simple model of this behavior, which is based on a neuronal network, okay? The neurons here, these are standard, you could do it in different kinds of cases. This was this was work done by Belén San Cristobal, mainly. The thing, in this particular case, we had Hodgkin-Huxley type of neurons, and we are connected in a more or less random connectivity. And the neurons are set up in such a way that they fire in a more or less regular manner, but the collective dynamics exhibits a more or less clearly defined rhythm. In this particular case, we are in a sustained awake state, if you will, but you can tune this system to operate in this type of up and down conditions that you will see in a minute. Okay, so the question is, what does this type of model predict? What happens when we increase the noise? Of course, in such a model, it's very easy to just play with it and say, okay, I'm going to increase the noise. But we want to do something that we want to make an experimental prediction. So the question is, what would control the noise in a real experiment? And what we found out is that the best way of controlling the noise is varying the excitability of the tissue. And we can do this by adding some source of extracellular potassium concentration, for instance. This varies the level of excitability. And you can see that as we vary this level of excitability, the noise measured in terms of what is the variability in the interval between spikes in these neurons, it really increases. So somehow this means that we can assume, we can interpret an increase in excitability as an increase in noise. We are not the ones that we're not the first ones doing this. So what we're going to do now is we're going to see what happens when we increase the excitability of the tissue that we can control in principle externally, okay? And then what we see is what we expected. If we vary the excitability at low levels of excitability or high levels of excitability, the irregularity is large. And when you tune the excitability, meaning that you get a more or less optimal amount of noise, you decrease the variability twofold. It's not much. Of course, this is matching what, well, you will see now. I mean, we could, theoretically, you could obtain much better results if you tune the parameters, but we wanted to be close to the experiments, and what the experiments say is that indeed this is the case. If you do experiments, and these were experiments done in Mavi Sanchez-Vives' lab, if you do experiments in cortical tissue, you see that increasing the excitability, which we could interpret as an increase of noise, of synaptic noise, you do reduce the variability at the beginning, and of course eventually you have too much noise and the system becomes irregular again. So in this particular case, we can interpret that noise is really helping you regularize the dynamics, and this has a physiological meaning. So that's all and well, but then the question is, what happens, this is electrophysiological, and with a very specific cell type. What happens now with more generic cell types, and what happens at the biochemical level? So now let me go back to more, if you will, classical systems biology type of questions, and I'm going to look at standard cell signaling in cells. And the question here is relevant, because if you think about it, cells are subject to a whole lot of different types of signals. Usually, in many cases, we are only interested in the response of the cell to one particular signal. And that's what many people study and what we have studied in the past. No? We study the response to a single signal in an isolated pathway. But what happens when you try to integrate all these signals, because you don't want to ignore the fact that you have all these other signals which are maybe more or less stochastic. You have some kind of chatter in the environment. This cell is subject to some signal, but then a whole lot of chatter. And the question is, what does this chatter do? Is this something, it is preventing the cell from operating and responding to this signal or not? Or is it because the simplest option would be no, the cell is actively making this other input silent, but this would be very costly. So maybe, I don't know, can the cell use this chatter in a positive way? So this is what we try to analyze with a theoretical model. In this particular case, we used a signaling network from a fibroblast. This was put together by the group of Gene Rogers at the University of Utah a few years ago. And they looked at hundreds of experimental papers in order to identify this network. And more or less, it's known what is even the logic at each node, the way in which all the other nodes act upon each of these nodes here. So the logic is known, the architecture of the network is known. We have identified, or they identified, a set of inputs that were biologically meaningful, and also a set of outputs, which we can identify, which we can relate with well-defined biological responses. So the question is, now, if we have noise in the inputs, what's going to happen? How does this network going to filter, how to modulate all this chatter that it's going to receive? So the first thing I'm going to show you is, let's assume that we have chatter everywhere, no special node, no special signal, and this is what we have. So now we have chatter everywhere. We increase the amount of chatter. Let me just like to point out that in this particular case, the maximum variability is at Q equal 0.5, because this Q is basically the probability of a node to be active or inactive. And we did this with a Boolean description of this network. So what we saw is the following. Interestingly, when you vary this noise, when you vary this chatter, what's happening is that these outputs respond differently. Some of the outputs are really responding in a non-monotonic manner with respect to Q. Some outputs are responding in a monotonic manner. And for different noise levels, some outputs or some other outputs are chosen. So here you can already see that noise can help you select which which outputs are going to respond to this particular state, which of course it's very noisy, okay? Now, it will be more interesting to see what happens when one of these inputs has some kind of meaning. So what we did is we took one of the inputs and basically added a signal. The simplest case would be just a periodic signal. And then our question would be, okay, how is the system responding to this specific signal? And you can see this is an example. If we look at the average response of all these outputs here, some of the outputs are going to respond to this signal, and you're going to see a nice oscillation at the same frequency, and some of the outputs are going to be irresponsive. The question is, how does this depend on the noise? And this is what you can see down here. When we vary the noise and we compute the power, the spectral power at the frequency of the input, that's the simplest thing that we can do here, you see that for certain levels of noise, you have a particular output that responds better to the signal, and this would be SAPK in this particular case, or for instance, for this other range of noises, you have P38. So this is already telling you that indeed noise can help you select which outputs are going to respond to a given input. Okay. We went even a little bit farther and tried to answer the questions where the information was going, how was it propagating from input to output. And we can do this by just looking at the correlation. So if we see, here the darker the lines means the information, the two nodes are sharing the information, and the more gray it means that the information is not shared so much. So really here the darker paths are indicating where the information is propagated through a network, and these are for the different values of chatter, and you can see that depending on the value of chatter, depending on the value of noise, you go in one direction or you go in the other direction. And in fact, you can quantify these type of things and say, and identify paths that are active for high noise, paths that are active for low noise, and they have, of course, different biological meaning. You can either have MAP kinase pathways, you can have CAMP, ACCAMP pathways, depending on the noise. So this is telling us that in fact cells, that's a proposal so far, that cells can use noise to route the information, not only to select the output that we have seen, but also to route the information. Another interesting thing that I would like to emphasize is that this type of behavior, interestingly, it's lost completely if we just perform random, if we do random surrogates of this network, and we don't need to randomize it too much. If you just randomize the logics that have been established by means of these experimental studies, we just randomize the logics. Even if you keep the structure, the architecture, the topology of the network, you're going to destroy this behavior. You're even going to destroy the simplest response to noise. These lines become completely flat if you do the simplest randomization of this network. So it's very appealing to think that this network here has been tailored by evolution in such a way that you can choose, you can take most of the profit of the fact that you have all this type of these noisy inputs acting upon the cell. Again, this is just a prediction, if you will. There's no experimental realization, and there are two reasons by which there are no experiments so far, in my opinion. One is that the cell type is very difficult. It's difficult to work with fibroblasts, and also that the circuit, the network, is very complicated. So one thing is that maybe we can try to look at a simpler model system, and we can try to look for also simpler circuits. And that's something else that we have been doing in recent years, and for instance, looking at the specific type of bacterium, which is Bacillus subtilis. In particular, we were interested in seeing how this organism responds to stress. And an interesting response to stress, in which we have been working on for several years already, is genetic competence, in which the bacteria become able to absorb DNA from the environment and try to survive in these conditions of stress. For us, what was interesting of this particular phenotype, and that's why we continue to look at it, it's because it's transient. You can see that it's a transient phenotype. Cells go in and go out. It's also stochastic. All of the cells are stressed, as you can see here by the green marker, but only this red cell is becoming competent. So it seems it is stochastically initiated, but once it is initiated, it stays there for a more or less well-defined period of time. So this is what we would call an excitable behavior. And now, if this is an excitable behavior, like the neurons I was showing you earlier, this means that maybe this system is going to be able to use noise in a very profitable manner, okay? So the first thing that we need to do is really prove that this is really noise-driven, and you can see that immediately, for instance, by looking at the fate of daughter cells. You can see this is the trace of the red marker, which indicates that the cells are competent. This is the same cell, and at some point it divides, and when it divides, the two cells, the two daughter cells, one decides to become competent and the one stays in wild type in vegetative conditions, okay? So you can really see that this is already a strong indication that, that this behavior is stochastically initiated, okay? Good, so we did many other things in order to prove this. For instance, another thing that you can do in order to prove that this is stochastically initiated is trying to control the noise. What happens if I now vary the noise in a controlled manner? And we can do it, for instance, we can do it globally. That's a nice way, if you have a bacterium mostly, in which you can vary the noise. And the thing is that what you can do is you can use a mutant, in this particular case, FTSW mutant, in which cells are not able to septate, but they are able to replicate their chromosomes. So this means that you are scaling up the cell. All the contents of the cells are scaled up. This means that you have more of everything, and of course you have less noise. So what this means is that as the cells become longer and longer, you have less variability, and you can see this very clearly here. This is just the expression of a constitutive promoter. As the length becomes larger and larger, the variability is increased. So the prediction is if there is less noise here, the probability of cells getting into this state, which is stochastically triggered, this competent state, is going to be lower. And indeed, that's what we saw by analyzing these movies from these long cells. As the cells become longer and longer, the probability of becoming competent went down. Okay? So this is another way of proving, if you will, that the initiation of this state is stochastic. Some other works by some other groups, for instance, the group of David Dubnow did another analysis. They control, instead of controlling the noise globally, they control it locally. They pick one specific gene, which is the master regulator of competence, and they varied in a well-defined manner the amount of noise acting upon this gene by varying in a balanced way transcription and translation. This is a method that was proposed by Alexander van Oudenaarde a few years ago in this paper. And they did this experimentally in this competence system. So for instance, if you increase transcription and decrease translation, what's going to happen is that you're going to reduce the noise. And this is what they did here, comparing the wild type situation with a low noise situation. And already here, just by looking at the plots, and they quantified it too, is that when you have a lower noise, you have a lower probability of initiation. So this indicates, again, that competence is noise driven. Okay? So we have here a circuit, and now I'll tell you how large a circuit is, a genetic circuit, that is using the intrinsic noise to do something, in this particular case, to transfer, to differentiate transiently into competence. But now, what is the circuit and what is the dynamics? How is this, I mean, can we predict something about how it's going to respond? The first thing is we had to decide how the system is. And there was some kind of discrepancy because, as I was telling you earlier, our assumption is that the system is excitable, whereas, for instance, David Dubnow and co-workers, they were claiming that the system is bistable. You just have two stable states, the blue and the gray, the competent and the vegetative, and the system just jumps from one to the other stochastically. Now, a bistable system and an excitable system respond to noise very differently. So we wanted to clarify whether this was bistable or excitable. And as you will see, the response is both, as usual. That's usually the response in life with no extremes. No? So you will see in a minute. So what we did is the following. We used the dynamical behavior, the dynamical character of this process to analyze statistically the outcome. Okay? We can define probabilities of initiation, probability of exit into competence, and probabilities of reinitiation after a well-defined period. And calculating the probabilities of the three events, we can classify the behavior of this system, and we can classify, for instance, into an oscillatory behavior, excitable behavior, bistable behavior, monostable behavior in which cells get stuck in competence or get stuck in vegetative conditions. For instance, if the probability of exit from competence is high, but the probability of reinitiation is also high, the system is going to be oscillatory. On the other hand, if the probability of exit is low, and the probability of reinitiation is also low, it's comparable to the probability of initiation, it's going to be excitable, because noise is going to just excite excursions from time to time, and you're not going to have periodicity, and so forth. So this means that now we can take our system, and we can perturb it. The idea is now we perturb it in a controlled way, and we classify the dynamics, and then we can try to see what is really happening here. And the good thing is that in our studies of this type of system, we came up with a circuit. This is a circuit. Okay? This is the master regulator which activates itself and represses the stress sensor. Okay? This circuit here, which is described by two very simple differential equations, can be perturbed. Of course, we can analyze the perturbation. We can perturb this circuit mathematically, for instance, varying these parameters here. But the nice thing is that we can do it also experimentally. Okay? So this input here is basically this parameter. This is the nutritional stress. And these two inputs, for instance, are corresponding to these two parameters. Now we can do is I can do two-dimensional perturbations of this in a systematic manner, trying to validate our model and therefore our interpretation of what's going on. So what we did, that's what we did. Now I'm going to show you in the next 3 slides two rows of results. This first row here is going to be a perturbation of these two parameters together, copy number, which is more or less a stress, and this constitutive signal. And in this other row, in the lower row, I'm going to show you, again, the stress together with this other constitutive signal. Okay? So I have two different pairs of perturbations in different locations of the circuit. Okay? And I'm going to try to see what first of all, what is the prediction of the model. We can do this doing bifurcation analysis. And this is the prediction of the model. Okay? The 3 figures here are basically the same. It's just in grayscale, I'm just plotting the 3 probabilities. And these 3 probabilities together, as I was telling you earlier, allow us to classify the dynamics into these different states. So in the particular case in which we perturb these 2 inputs, okay, what we have is that we have 2 dynamical regimes, the ones that I was telling you earlier, either excitability or bistability. If we perturb these 2 inputs, which are more separate, more apart from each other in topology space, if you will, what we have is all 4 dynamical regimes, okay? And we identify them by looking at the behavior of these probabilities, and you can calculate the bifurcation boundaries and so forth. And the question is, what is the experiment telling us? So I'm going to show you what we see from the experiments. These are time traces that they were computed painstakingly by Lorena Espinar, who probably is around here, I hope. And you can see here that there are the two behaviors, excitability and bistability, in the case in which you perturb these 2 inputs. Okay? Bistability is clear because you see that some cells stay vegetative and some cells go competent and stay competent. Okay? And then you have excitability here, which is what we assume that the system is operating in this regime. And on the other hand, you have, if you perturb the other 2 inputs, you have all 4 dynamical regimes. So you can already tell that more or less we have a good agreement. But the agreement is not only qualitative. We can see that it's quantitative because we can really, using well-defined controls, we can quantitate what are our inputs in our experiment, and so that we can now place the corresponding experimental results on top of our parameter diagrams, and we can see that the agreement is relatively good. Okay? There are some caveats that we could discuss about this. How do you define the different regimes? But the agreement is in general good. So this means that, first of all, our model so far seems to be more or less reasonable, more or less correct. And we assume that when we are in an excitable regime, we are in this area here. Whereas maybe when David Dubnow says that they are in a bistable regime, maybe they have a little bit more stress. So our conclusion is that depending on the stress level, you can be in one state or the other. And this means that everybody's right and everybody's happy. And on the other hand, this means that depending on where you are in the phase diagram, you're going to have one response to noise or another. Okay? So now, let's consider that we are operating in an excitable regime. For some conditions, the system is excitable. But now, there's another question, which is you can get excitability in different ways. For instance, this is the way in which we get excitability, I showed you earlier. And this is the classical way. This is much more common in nature, even. Okay? So the question is, why did nature choose this instead of this? So we have a problem of degeneracy here, similar to what Mark Isalan was telling yesterday. So you have all these different circuits that produce a stripe. Which one is being selected? So in this particular case, we wanted to address the question of why this and not this. And to our surprise, the noise was involved in the answer. Okay? The question is the following. Well, in order to investigate this, what we did, mostly Groswell in his lab, was rewire the original circuit and convert it into this circuit. And we can do this. I'm not going to get into details. This is another version of our original circuit. And we can rewire by getting rid of this negative feedback loop and getting another feedback loop with the polarity reversed. Okay? Because the only difference between these 2 circuits is the polarity of these 2 interactions. So we just switch the polarity of the 2 interactions, and then now we get another version of the circuit which has a different architecture. We tune it using also mathematical model, and we can really make the 2 systems, well, of course, this we know, but we can make the synthetic system operate in a behavior which is similar to the native system. Not only from the point of view of what is the dynamics of the expression of the protein, but also physiologically, okay? We checked, and we saw that physiologically, this behaves as a competent system would behave, okay? You can uptake DNA and can survive and so forth, okay? So this state is synthetic, but it's physiological. And now we can ask the question, is why one and not the other? So then what we saw immediately when you analyze time traces of these guys is that the native circuit, I told you earlier that the durations were stereotyped, but they are not perfectly stereotyped. In fact, it's a bit noisy. The durations are a bit noisy. The cell goes up, and it goes down, but the time at which it goes down can vary a lot between 10 hours and 40 hours. On the other hand, interestingly, the synthetic circuit, the one based on the other architecture, was much less noisy in terms of durations. So now now it's intriguing. So there is a difference between the two systems. One is much more noisy than the other. The natural one is much more noisy than the other. Why can it be? Maybe this is an answer why the system selects one or not the other. So that's what we try to understand. And in order to understand we realized that, of course, we have to take into account the fact that being in competence has both a cost and a benefit. The cost is that while you are in competence, you cannot grow so quickly because you have to do other things. So you have to grow more slowly, and of course this is a cost, especially if you are under stressful conditions. The benefit is that, of course, the longer you are in competence, the higher the probability you're going to have to uptake some DNA that might help you. So you want to be for a long time in competence, but maybe not too long. And on top of that, you have to take into account the fact that, of course, it's not clear when the cell is going to find a molecule of DNA, because it's not that this DNA concentration is going to be there at large levels. Okay, this DNA is coming from other cells that have died probably, and this is not going to be so common. So the question is, okay, taking into account these two things that have to be balanced, and the fact that you have an uncertain environment, how does the system cope with this? Okay. So what we did, again, we all did in his lab, is this type of experiment. What they did is, okay, let's vary the amount of DNA. Let's go from very little DNA in the medium to very high DNA in the medium. Let's use some assay in which cells can only survive if they uptake DNA, for instance, in the presence of an antibiotic. And let's analyze the output of the cells in terms of how many transformants, how many colonies you have after adding the antibiotic. Okay? Now, if you think about it, the native system is very broadly distributed, and the other one is very sharp and with a short duration. Now, if you have a very well-defined duration, something is going to happen, which is the following. If you have a lot of DNA, it doesn't matter. But as soon as you reduce the DNA, at a certain point, you're going to have a very bad response, because you don't have a lot of variability. As soon as the DNA concentration is low enough so that you have to wait more than this time in order to take up some DNA, if you are not waiting long enough, you're going to die, basically. So this means that our prediction is that the artificial cells, the synthetic cells, are going to go down much faster when the concentration of DNA decreases. And that's indeed what we saw. Here you have a comparison of what happens when you decrease the amount of DNA between the natural system and the synthetic system, and you can see that really the synthetic system is much less able to cope with a decrease in DNA than the natural system. So this means that our interpretation is twofold. First, that competent systems, this competent circuit is basically generating randomness in order to be able to survive under conditions in which are not very well defined environmentally. Okay? And the second one is that you have two types of circuits here that somehow cover a different range of possibilities. For instance, the native circuit is very good in terms of response range, because the system responds very well to a wide range of DNA concentrations, but it's not very precise. Okay? And so this happens when you have a lot of noise in the negative feedback loop, which is basically what causes noise in the durations. Okay? On the other limit, if you reduce the noise, you have a system which is very precise, but the response range is low. So this means that there is a trade-off between response range and precision, and in fact, in nature, in some cases, selects circuits that are responding to this type of conditions and other systems that respond to this type of conditions. In fact, most circuits behind the cell cycle and circadian clock, the core of the circuit is an activator repressor module like the one that we have in our synthetic system. Okay, so now to finish, I would like to address another issue. I mean, of course, this is, again, very interesting what happens with more complicated cells. Can we learn something about how complicated cells use noise? And then we move to the field of pluripotency in embryonic stem cells, which is something, pluripotency is a very interesting phenotype because it is very variable. I mean, this has been established for many years already, and you can see it if you look, for instance, at the expression of a certain pluripotency factor such as NANOC. You can see a huge variability in the expression of this pluripotency factor cell to cell, okay? Interestingly, I mean, if you look at the distributions, for instance, you see very broad distributions, sometimes multimodal distributions, so you can really determine populations which are high in these pluripotency factors and low in this pluripotency factor. And another interesting thing is that this is not a static population, because if I now select subpopulations from here using FACS, for instance, now I select this population here, and I let it go, and I culture it for a while, and I select this subpopulation in the middle, or a subpopulation of high NANOC cells. All these subpopulations are going to recapitulate after a few days or a month the initial distribution, okay? So this really means that this is an attractor, and all the elements, all the cells in this attractor, or most of them, are basically moving back and forth. In fact, it's not that true, because cells in low NANOC are more prone to differentiating, and some of them are going to be lost. But still, after 1 month, at least particular conditions, you reproduce more or less the original distribution. So there is a lot of dynamics here and a lot of variability. And the question is, what does this mean? How do we interpret this? Initially, a few years ago, since we were coming from this paradigm of excitability, we interpret this in terms of excitability. We came up with a model involving NANOC and another of the main key pluripotency factors. And we came up with a very simplistic model again in which we would interpret these excursions into low NANOC states which are more prime for differentiation in terms of excitability, okay? But after a while, what you have to do is, of course, if you have to validate or try to validate your models. There was some observation that this model could not reproduce, which is this one, okay? It's an interesting observation, which is when you look at the correlation between noise in different factors, what happens when you not only look at NANOC, but also at NANOC and NOC4 together in the single cell level. And you can see that each point here is a single cell. And you can see that the correlation is a bit weird. I mean, there is a boundary here below which you find no cells. That's really intriguing. This is reproducible under different conditions. You repeat the experiment many times, and you can see a very well-defined line here, and there is a forbidden zone. So the correlations are not trivial. It's not that the cells are complete, that NANOC and NOC4 are completely correlated, not at all. But it's not that they are completely uncorrelated either. So there is a very strange correlation here. And then the question is, we could not reproduce this with our excitable model, so what's really going on with this variability? Okay, so what we did is the following. After a while, we came up with a potential explanation, and the explanation is very simple. It involves the fact that since you have some sort of correlation, this means that there has to be some sort of interaction between NANOC and NOC4. And in fact, it's known that NANOC and NOC4 form complexes. So let's assume that we have a simple protein-protein, the simplest possible protein-protein interaction circuit like this. Okay. So we take this thing, NANOC and NOC4 form a complex, okay? Of course, now this thing is going to be fed by noise. I mean, because of course NANOC and NOC4 are going to be expressed continuously, so we're going to have some transcriptional noise. And this is where our noise in this particular case enters, okay? So we have a protein-protein interaction network, very simple in this case, which is somehow modulating the noise that gets into it. Now, this extremely simple circuit already has a very interesting behavior, which is the following. If you analyze it mathematically and build a mathematical model of this, the fact that you have this complex together with an asymmetry, which is that NANOC is more unstable than NOC4, which we don't have direct experimental evidence yet, but we have indirect experimental evidence. We can get into this later if you want. This asymmetry, together with this complex, lead to a situation in which the system can only live in this portion of phase space, which is this triangle that I was telling you about earlier. Our interpretation is that noise is kicking the cell out of this line, and then it does some excursions in this area, and this is where we see the cells when we do the immunostaining. And then immediately the cell tends to the slow manifold, and then it keeps going down. So it's not written here, it's not shown here, but what cells are doing they are going up, they're going to the right, and then they go slowly down. And when they are in this line, they are in pluripotent conditions. So our interpretation of pluripotency is that we have cells living in a slow manifold, which is fed by transcriptional noise. Okay? So this transcriptional noise is maintaining the system in a poised state in which you can have cells which are pluripotent and cells which are ready to differentiate. Okay? So the question is, can we really reproduce, can we validate this model? So we found out that, for instance, we wanted to validate what is the response of these pluripotent cells to other signaling states. And one of the conditions that we wanted to understand is the so-called 2I conditions. These 2I conditions are the following. If you add 2 specific inhibitors to the growth medium, you're going to enhance a lot pluripotency. Okay? Your cells become very strongly pluripotent under these 2I conditions. Now, in order to reproduce these results, we had to enlarge our circuit, which was here before. We had to include, for instance, beta-catenin, for instance, and TCF3, because they somehow mediate the 2 inhibitors that are affecting these two, that are affecting the circuit via these 2 pathways. Okay? So now we have a slightly more complicated, but not too complicated protein-protein interaction network that is filtering some kind of transcriptional noise. And what we can see from this network, I'm not going to get into details because I'm already running out of time, that we can reproduce the correlations that we observe experimentally. These are the correlations that we see from the model. In 2I conditions, by the way, the system is much more correlated along this slow manifold, both in the experiment and in the model. And this somehow supports our contention that it is the manifold what's really characterizing the pluripotent state. In 2I conditions, all cells live in this area. And basically, you have no cells around here, and these are the cells that are poised for differentiation. And again, this is driven by noise. And we can also explain different kinds of mutants. Okay, we can explain all kinds of mutants. This is, for instance, a comparison of all the different correlations that we have been able to measure with the immunofluorescence between all the factors in normal conditions, serum plus leaf, and in the pluripotency enhancing conditions, 2I. And we can reproduce with the model this type of correlations. We can also reproduce of all kinds of mutants. And finally, one thing that the model tells us is something interesting, which is if we compare them, at least the model is able to tell us all the different components of the pluripotency network, where are they? If they are in free state or if they are in the complex, for instance, in different complexes that this network has. And one of the things that we get when we compare this with the normal conditions with the enhanced pluripotency conditions, is that, for instance, in the normal conditions, you have a fraction of cells which have free OCT4, whereas in the enhanced pluripotency conditions, you have no OCT4 at all. So this somehow led us to think or to conjecture that what's happening in pluripotency is that basically you don't want to have too much OCT4 which is free, okay? And in fact, that is the case in the proposal that was already made long ago, in which basically this was from Austin Smith, in which he was interpreting what pluripotency in terms that OCT4 has to be in a certain range. If it's too high, you're going to lose pluripotency, and if it's too low, you're going to lose pluripotency. And in fact, what we are seeing is that you cannot have too high OCT4, because if you have too high free OCT4, you're going to have differentiation. Now, let me emphasize again that these interpretations come from a model, a protein-protein interaction model, that is using transcriptional noise. And this transcriptional noise, what it's allowing the cells is to somehow bed hedge, because the cells, thanks to the noise, can move up and down this manifold and can get out of it in such a way that some of the cells are going to maintain its pluripotency conditions and some of the cells are going to be poised for differentiation. So in this particular case, noise here is also allowing the systems to do some kind of bed hedging. Okay? So I think that is all. I'm going to stop here. So what I have shown, I have tried to show that there are multiple types of biological control networks that can profit from noise. I have talked about cortical neuronal networks, signaling networks, gene regulation networks, and protein-protein interaction networks. And its particular profit, it can happen in many ways. For instance, noise can be a rhythm enhancer, noise can be an information router, or noise can be used, for instance, in the case of competence or in the case of stem cells, as a bed hedger, because it allows the cells to be ready for many different kinds of situations that it can find. So let me thank all the people who have been working on this, my PhD students, Lorena Espinar, Marta Diaz, Paul Roy, and Belén San Cristobal. 3 of the 4 are already out. Lorena is now working at UPF, Pau is in Cambridge, and Belén is at CRG now working with Mara Diersen, and we're continuing our collaboration with her in these up and down dynamics things. Thanks also to Tony Pons and Nuria Domeder, who were my former postdocs, and all the experiments, most of the experiments have been done in collaboration with Tolga Chagatai, Groswell, group of Alfonso Martinez Arias, Silvia Muñoz did most of the experiments in stem cells, Mike Kerelovitz, and Mavi Sanchez Diaz. Thank you very much for your attention.